Hi class, this is Jeff again. We're on lab 3, or we're going to start on page 6. And so it's going to be the microscope. Alright, so let's introduce you to some of the parts of the microscope. Okay, so these are the oculars, or eyepieces, but we call them oculars. And they have a magnification power of 10. And so if you look at this, there's a 10x, and so that means it's going to magnify 10 times, and that's will be significant a little bit later. The eyepieces have an adjustment to fit the width of your eyes. Okay, so we can do this, move them in and out. Okay, and you'll just have to fix it so that when you're looking through this, you don't see two circles, you see one circle. Okay. This is the base. Down here, this is the arm. This is the nose ring. Okay, and the nose ring has below it 4 different lenses. They're called objective lenses. Okay, and they have a multiplication factor. So they have, the shortest one is a 4/0, and then there is a 10/0, and then a 40/0, and then a 100. Okay, so that they stand for the magnification power. So this 4 one will magnify it 4 times. So coupled with the ocular of 10 times and 4 times, we multiply those together to get the total magnification. So this is going to be 40 times magnification. 10 for the ocular, 4 for the objective. So that's 40 power, we call it. So that means it magnifies the image, makes it look 40 times bigger than it is. Okay, then we go to 100. That's 10 times a 10, which is 100 power. Okay, then we go to the 40. That's going to be 10 times 40, or 400 power. And then we got to go to the 100 lens objective. So that's 10 times 100, or 1,000 magnification. So it magnifies it 1,000 times. And we have to use the 1,000 power magnification, or the 100 lens, in order to visually see the bacteria with any detail. Okay, so they're very tiny. And okay, so that's the objectives, and then the ocular. There is a knob over here, right here, that we can twist and it unlocks, and then we can rotate the oculars. If we're in class and I'm helping you out, you'll say, well, I can't find this, or would you, oh, this looks pretty neat, would you look at it? I generally won't come around and look into the eyepieces here. I will be on the other side of your desk, and so I'll loosen that up and spin it around and look at it this way. Okay, and then put it back, but just make sure that you lightly tighten this when you're done. Okay, so other parts of this is the stage area. They used to have a separate stage and then a mechanical stage. Now we just call this the stage area. Here's a slide holder. So the slide, the microscope slide, so here's the microscope slide. We'll separate it or stick it in the clip. Okay, so this clip right here opens up, and we clip it, and this slides down and holds it there. Okay. Below that is the condenser. Forgot it for a minute. And the condenser will magnify, or will shape the light to a point, and it'll come out right at the specimen on the slide. Underneath the condenser is a lens shutter, and so that'll be like a bird's eye lens, and it'll open up and close. So it closes like that and opens up. Okay, and I'll take that out here and let you see that. Okay, so if you look closely, when I move this lever, that opens up. So that's the iris diaphragm there. And then the lens part is the condenser. So the light will come up, and if you have a very high magnification, you need a very high source of light. So one thing you can do to make that better is open up the iris diaphragm to let more light in. Okay, and it is a matter of opening and closing this to get the contrast for the bacteria that you're looking at. Okay, we'll put that back up here. It has a little set screw that we might want to make sure that it's ready to go. Down here is the light source, and off to the side is a dial right here, and everything's backwards on this scope, or on this video, I think. And so you want to go to the right side as you look at the scope, and there's a dial here. Okay. And so we're going to take that and we rotate it, and it makes it brighter. See, you can see the light getting brighter or dimmer. And generally, the higher power, so we want to have more light. Okay, so I like to have students just crank that up pretty high, and then we'll adjust the light with the condenser. And there is an adjustment for the condenser right here. There's a knob, and we can lower and raise the condenser. So the condenser was right here, and we raised it up and lowered it. Now we're raising it back up. So as you increase the magnification, you want this to come up. So when we're on oil lens, or 100x lens, the objective, we want this all the way up. Okay, because the more magnification, you need more light. It gets too dark. Okay, and so we'll bring that up, and then we can adjust the iris diaphragm lever right here. All right, so that's the light sources and how to control the light. So in recap, there's 3 places to control light. One is the original light source, and there's a rheostat or dimmer dial onto the side. And then there is the condenser, which we can raise and lower. Raise, higher power, higher, we raise up the condenser. And then there is the lever that opens and closes that bird's eye mechanism. So this, as we get higher magnification, we want more light, so we open it up and it gets more light. Okay. All right. And then once we have our slide on there, we want to center it. And then to move the slide, there's these controls on the side. They're right here. So one of them goes front and back, so the top one goes front and back, and then the bottom one goes left and right. And we use that to center our sample right into the center of the light. And we can see the light coming up through the slide, and we just want to make sure a sample is visible in that light area. All right, and then we have the focus. So we have the coarse focus is the big knob right here. It's the inside one. And so we raise the stage up and down. And then the fine focus is the smaller one here, right here. And that just does minor adjustments.